Welcome back to Unit 3, Part 2. Here we're picking up with endocytosis and exocytosis. We were talking previously about bringing things in through active transport and passive transport. Now we're going to talk about bringing bulk items in, so a lot of things coming in. And we're going to do that through endocytosis, or we're going to get rid of large amount of product through exocytosis. So many molecules are just too big to get across the cell membrane using that passive or active transport. In order to bring these things in, we have three different types or three different ways to bring them in. We have phagocytosis, pinocytosis, and receptor-mediated endocytosis. Now with phagocytosis, this is going to be bringing large amounts of solid products in. Pinocytosis is going to be bringing liquids in large amounts. And then receptor-mediated endocytosis means it's actually going to hook into a receptor, kind of like what we saw with adrenaline having to hook into that specific protein at an active site. And it's going to use that receptor protein in order to be able to bring itself in through a vesicle. And so here, looking at the top picture, you're going to see that's an actual cell bringing a large molecule into itself. And it's going to do this the picture below is going to show you a solid particle. And so this is going to be through phagocytosis. That particle is going to bump up against the cell membrane. As it bumps up against the cell membrane, it starts to push in on the membrane, creating a vesicle or kind of like a cell membrane around that product it's bringing in. And then once it comes in, it makes that perfect vesicle around it. So pinocytosis, like we said, that's cells taking in dissolved particles and liquids, so it's kind of like referred to as a cell drinking. And then you have your receptor-mediated endocytosis, where the particle is going to come in, bind with specific receptors there. In this case, it's going to be cholesterol. And so that cholesterol is going to come attached to those protein receptors. Once it attaches, it's going to allow it to come in or be pulled into the cell. It still creates a vesicle around it, and then it's going to take it to the part of the cell that needs it, and it's going to break it down. Faulty cell membranes are going to be a primary cause of cardiovascular disease. So what modification to them might be an effective treatment? So looking here, that's referring to things such as arteries. With this artery, you see heart disease is going to be formed on the right side of the page. A healthy artery is going to be on the left side here. As you start to take in LDL, which is going to be referred to as the bad cholesterols, it's going to start to cause the buildup or blockage of cholesterol in that artery. And so that lipid that's going to form there is going to prevent blood flow from going through the artery. Eventually, it will become completely blocked. And that's going to give you risk of cardiovascular disease, or it could give you risks of strokes, heart attack, things like that. So here you see white and red vesicles in the pancreas of the cell that contain hormones, including things such as insulin for secretion into the bloodstream. And so exocytosis is going to be used to secrete these. So you see it's put into a transport vesicle. It's done here at the Golgi apparatus. These molecules are then going to make their way towards the cell membrane. And when they get to the cell membrane, they're going to push up against the cell membrane. The vesicle is going to fuse with the plasma membrane, and it's going to push those particles out to then travel to other parts of the body. So when molecules can't get into the cell through diffusion or a pump, if they're just simply too big, cells can engulf the materials with their plasma membrane in the process called endocytosis. Similarly, molecules that need to be taken outside of the cell to other parts can be removed or taken out of the cell through a process called exocytosis. In both processes, the plasma membrane moves to surround the molecules or particles and forms little vesicles that are going to be pinched off inside of the cell or fuse with the plasma membrane and dump the contents into the outside portion of the cell. Connections between cells are going to hold them in place, and they're going to enable them to communicate with each other. It's going to involve numerous types of proteins and glycoproteins, or adhesion molecules, that are going to put them together. So three types that we're going to talk about in this chapter or unit are going to be tight junctions. These are going to form between cells to make waterproof tight seals. So it's kind of like caulking around a tub. It's going to hold them together tightly. They're very strong. You would want to find these in places such as the stomach or the urinary bladder, where you don't want the contents to leak out. Desmosomes is going to be another type. They're going to act as Velcro and fasten things together. So think of fastening cells together in the skin. You want them to have a strong connection, but you also want them to be flexible. And then the third type are going to be gap junctions. These junctions are going to be able to act as tunnels or passageways between cells that hook cells together. This is going to allow materials such as cell signals to go from one cell to the next, or even things such as electrical signals in a heart contraction. So tight junctions, like we said, they form continuous watertight seals around cells and anchor the cells in place. Particularly important in small intestines where digestion occurs, also in the stomach and in the bladder. Desmosomes are going to be like spot welds or rivets that fasten cells together into strong sheets. They function like Velcro, holding the cells together, but they're not going to be watertight. This is going to be found in much of the tissue lining cavities and of the animals' bodies. And then you have your gap junctions. These are going to be pores surrounded by special proteins that are forming open channels between two cells. With these types of junctions, they're going to be important in cell-to-cell communication. So how can a lack of communication between cells lead to cancer? Well, most cells, once they start to grow and divide, they're going through mitosis, and once they feel like they're filling up a space, they come in contact with each other, they have a form of contact inhibition. So if they become too closely packed or they get close to another cell, then they stop going through mitosis, they'll stop growing, and they keep their structure. However, cells that are cancerous that start to grow out of control do not have this contact inhibition, so they'll continue to grow. And they start to pack those cells tightly together and they form a mass, and that mass is referred to as a tumor. Plasmodesmata is going to be a tube-like channel that connects cells to each other and enables them to communicate and transport between each other. So consider a plant as one big cell. How would you do that? How would they get information from the top of the plant to the bottom? And it's going to be done through these plasmodesmata. Is a breakdown of cell-to-cell communication related to cancer? Well, it is, mainly because we discussed that contact inhibition. They don't have contact inhibition like normal cells do, so they'll continue to grow, and that continued growth forms tight masses of cells that don't perform the function that that cell is supposed to perform. They simply just continue to divide and divide until they form a huge mass called a tumor. That tumor eventually starts to use up the energy that would go towards cells that actually function to help the body. And so it's going to starve those cells while fueling the cells that aren't performing a proper function, which are the tumors. So in summary, multicellular organisms, most cells are going to be connected to each other. The connection can form a watertight seal between the cells, which are tight junctions. They can hold sheets of cells together while allowing fluid to pass between the cells, desmosomes, or they can function like secret passageways between the cells that allow the movement of cytoplasm, molecules, and other signals. These are going to be your gap junctions. Some cells are going to be connected to each other and be able to pass information throughout the entire body, such as plants, being able to communicate with each other, and that's going to be through their plasmodesmata. There's going to be 9 important landmarks that distinguish eukaryotic cells, and of course these are going to be your organelles. So the nucleus is going to be the first one we talk about. That's the cell's genetic control center. It's going to be the largest and most prominent organelle in most eukaryotic cells. Remember we talked about the plants, sometimes that large central vacuole is larger than the nucleus, but for the most part you'll find the nucleus. So with that nucleus, it has that genetic information, and it stores the heredity information that that cell's going to need to make things such as proteins and function properly. So looking here, you will see that nucleus in the center is going to be in purple. Inside the nucleus, you're going to have the nucleolus. That's going to be where ribosomes are made. You see it's porous within that nucleus. And so the ribosomes will leave through those pores, the nuclear pores, and go out into the cytoplasm to actually function. Also going around the nucleolus, you'll see the DNA, which is going to be in a form of chromatin during its interphase or natural life cycle. And then when it gets ready to divide through mitosis, it's going to condense down into chromosomes. Looking at the cell membrane that goes around the nucleus, which is going to be referred to as the nuclear membrane, it's not just a bilayer, but it's going to be referred to as a double bilayer. So notice that it actually has 4 layers here. And then, of course, it's going to have these pores. So it's a double phospholipid bilayer, which in total means 4 layers. Chromatin, like we said, is going to be a mass of long, thin fibers consisting of DNA with some proteins attached. The nucleolus is going to be in the center of the nucleus, and it's responsible for assembling ribosomes that it then sends out through the nuclear pores into the cytoplasm. Those ribosomes are going to act as little factories. So in summary, the nucleus is usually the largest and most prominent organelle in the eukaryotic cell. It directs most cellular activities by controlling which molecules are produced and at what quantity. And the nucleus is the storehouse for the hereditary information. Cytoplasm and cytoskeleton in a cell are going to help with its internal environment, physical support, and movement. So the cytoskeleton is going to be made up of 3 chief purposes, or 3 primary things. It's going to have microtubules, intermediate filaments, and microfilaments. So it acts kind of like the scaffolding or like the skeleton of the cell. It helps provide shape and support. It controls the intracellular activity or flow and enables movement inside the cell and of the cell. So with the microtubules, they're going to be the thickest tubules there. They are hollow tubes. They make tracks to which molecules and organelles within the cell may attach themselves to and move them throughout the cell. Intermediate filaments are going to be the middle-sized filaments. They're durable, they're going to be rope-like, and very numerous and overlapping. And these are going to give cells the great strength that they have within. Next is going to be the smallest filament, and that's referred to as your microfilament. They're long, solid, rod-like fibers, and they help the cell contract during cell division. So you're going to see these during mitosis. Cilia and flagellum are going to be 2 structures that you find outside of the cell. Cilia that you see on the left-hand side are going to be hair-like structures that help move things over or off the cell. So lots of times you'll have mucus along with this cilia, and you can find it in places like the respiratory tract. So it might capture microorganisms and work in a wave-like motion to move that bacteria or microorganisms out of the respiratory tract. You can also find it in the uterine tubes, where it helps move the egg along to the uterus. Now with flagella, you're only going to find that in animals or in humans in the sperm cell. And it's going to work in a beating-like motion or a whip-like motion in order to move it from one place to another. So in summary, the inner scaffolding of the cell, which is made up from proteins, is going to be called the cytoskeleton. And we saw it was made up of 3 different types of fibers: the microtubules, intermediate filaments, and microfilaments. It's going to give animals' cells their shape and support. It helps them have the ability to control movement, and it's going to serve in a series of tracks which organelles and molecules are guided across or around inside of the cell. Next, going to our organelle, which is referred to as the mitochondria. This is going to be the cell's powerhouse. It's shaped kind of like a kidney bean that you see here, and often referred to as a sac within a sac. Its function is to act as the energy converter inside of the cell, and it harvests energy to be used in cellular functions. Now looking at it, notice it has the circular DNA, and remember we referred to as that circular DNA being found in prokaryotes. Remember, they think that the mitochondria formed when the cell many millions of years ago engulfed a bacteria, and they work together in a symbiotic relationship, and that's referred to as endosymbiosis, where it brings it in and then they work together. So the mitochondria, remember, has its own DNA, and it's going to have the DNA of your mother. So it won't match up with the DNA inside of your nucleus. Cells such as muscle and liver, which have use a lot of energy, are going to have up to 2,500 mitochondria per cell. It also has a intramembranous space in a matrix, and lots of times is referred to as a sac within a sac. So we just talked about this endosymbiosis, where they think it may have existed separate as a single-cell bacteria-like organism outside of that cell. And then it was engulfed into the mitochondria and then started to work together with that mitochondria. We all have more DNA from one parent than the other. I know that lots of times you hear that it's 50-50, but this technically isn't true. So who's the bigger contributor, mom or dad? Well, it's going to be mom. Why? Because of this mitochondria. Remember your mitochondrial DNA, or if you had a DNA test, it'll be called the mtDNA. This actually comes from mom, so it will match up with your mother's DNA. So you have slightly more DNA from your mom than from your dad. So in summary, the mitochondria is going to be the energy contained within the chemical bonds of carbohydrates, fats, and protein molecules is going to be converted into carbon dioxide, water, and ATP, and the energy source for all cellular functions and activities. Mitochondria may have their own evolutionary origins as a bacteria that came in and started to work with that organelle. So fat cells and cold temperatures. They're going to use 15 cats from 5 litters to study this. From each litter, one cat is going to serve as control, while the other cats are going to be exposed to extreme cold temperatures. One's going to be temperatures of 30 degrees Celsius for 2 periods of 1 hour per day. The rest of the time, all these cats are going to be kept at 20 degrees Celsius. So why did researchers use multiple cats from each litter? Well, they want the same litter, so they have the same genetic makeups, and they're going to be close relation. And then they want to be able to compare them to each other to see how it affects them. So after 1 week of this treatment, the team collects samples of fat tissue from all of the cats, which they examined microscopically. In the fat tissue, they're going to measure the number and size of mitochondria, the number of tiny blood vessels or capillaries per cell, and the size of the cell. So what was the purpose of counting the capillaries? Well, capillaries are going to be responsible for bringing oxygen and nutrients to cells and taking away carbon dioxide and waste products. So it's going to, if they have less of these cells or the cells shrink, they won't need as many capillaries. Cold stress is going to cause a change in these fat cells. So in the animal cells exposed to cold, the mitochondria become more numerous because they're going to have to burn a lot of energy in order to keep the body going when it's extremely cold. So each is going to triple in size because we need more energy. The more energy burned, the more heat released, keeping that animal warm. The number of blood vessels is going to double because we have to bring more nutrients here in order to feed these cells. Also, you're going to need more blood vessels to take the heat away to warm that body. The fat cell size was going to be reduced to less than 1/4 of the size found in the cells in the control group of the animals. The reason for this is because it's using those lipid droplets in that adipose tissue in order for it to burn as energy. So looking at the table, why do you think fat cells in the cats exposed to the cold shrink so much? Well, like we said, essentially it appears that the cells in the fat tissue upgraded their heat generating systems, and in order to do this, it's going to have to use that lipid storage, which was found in these cells, as its energy source. So it needed more energy, and it got it from those fat cells. So in summary, form follows function in an organism's cells and reflects their environment. When cells must perform intensive heat production, they significantly increase the number and size of their mitochondria. They also increase the blood supply to the tissue and make existing stores of use existing stores of energy. Moving on to our lysosomes. These are going to be the cell's garbage disposals in sense. They're going to function as floating garbage disposals that digest and recycle cellular waste products and consume materials. When old worn-out cells or cell parts become non-functional, the lysosome will then engulf them and destroy them, break them down, and reuse what materials that it can reuse. So they are membrane-bound, they're going to contain digestive enzymes and acids, and they're going to break things down. So they're round, membrane-enclosed, acid-filled vesicles that function as garbage disposals. So why is Tay-Sachs disease like a strike by trash collectors? It's going to occur as a genetic mutation with 50 different enzymes that are necessary. So malfunctions sometimes occur in these. And like we said, it's a genetic disorder. So individuals inherit an inability to produce critical lipid digesting enzymes, so they can't digest fats. Lipids continue to be sent to the lysosome where they accumulate and digested. This is going to lead to a sort of lysosome constipation, so it starts to fill up with all of these fats, but it's not able to break them down. They swell until they finally burst. Remember, that's called lysing. And then it digests the whole cell until it chokes the cell to death because those enzymes start to break down other things in the cell, but not these fats. It's going to be fatal within the first few years of life. In summary, lysosomes are going to be round, membrane-enclosed, acid-filled vesicles that function as a cell's garbage disposal. They're going to be filled with about 50 different digestive enzymes, which enable them to digest all different types of materials. So they can dismantle and break down these macromolecules and be able to reuse some of those, but they also can digest and break down disease-causing bacteria and even viruses. Next, we're going to the endoplasmic reticulum. And so these are going to be two different types. You have what's referred to as the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Along with these membranous structures, you're also going to find the Golgi apparatus. So the functions: to produce modified molecules to be exported to other parts of the organism and to break down toxic chemicals and cellular byproducts. So looking here at the rough ER, that's what you can break it down to if you don't want to put endoplasmic reticulum. Their function is to modify proteins that will be shipped to other locations in the endomembranous system and the cell surface or the outside of the cell. So it's always usually attached or around the nucleus of the cell. It's going to have these dotted structures, which give it its name rough because it looks rough. And those are simply going to be ribosomes that are attached to it. So the ribosomes will create the proteins, and then they get modified by that endoplasmic reticulum. Once they've done that, then they will go to the Golgi apparatus for export. Next, you have your smooth endoplasmic reticulum. This one is going to help synthesize, which means to produce or make, lipids such as fatty acids, phospholipids, steroids. It's also going to be responsible for detoxifying molecules such as alcohol, drugs, and metabolic waste. So the more smooth ER you have, the better your body is at breaking down chemicals and things that you bring into your body that could be toxic. So let's say that you drink a lot of coffee. That caffeine in the coffee or in sports drinks or anything like that, or even alcohol, those substances are considered to be drugs or toxic to the body. The smooth endoplasmic reticulum are going to be able to break those down. So if you drink it quite frequently, your body's going to create more smooth endoplasmic reticulum within your cells in order to break these down very quickly. This makes things such as caffeine, alcohol, or even drugs not last as long, so this builds a tolerance for your body. If you don't expose yourself to these a lot, then when you take them in, it takes very little in order to affect your body. So how can long-term use of one drug increase your resistance to another, different drug that you've never encountered? Well, let's say you drank a lot of coffee, or maybe you take a lot of Tylenol. If you do that, then your body already has a lot of these smooth endoplasmic reticulum. So when you take a different type of drug, it still does the same function. It breaks them down. So you already have a lot of smooth ER to do that. So it might take more painkillers, more anesthesia, more of a different type of drug in order to affect you because your body's already very efficient at breaking these things down simply because they already have a lot of endoplasmic reticulum there. So in summary, the production and modification of biological molecules within the eukaryotic cells occurs in a system of organelles called the endomembrane system, which includes, among other organelles, the rough and smooth endoplasmic reticulum. The rough ER, proteins are going to be created and then shipped elsewhere in the body, then folded and packaged. In the smooth ER, lipids and carbohydrates are synthesized And alcohol, antibiotics, and other drugs are going to be detoxified and broken down. The Golgi apparatus is going to be another one of your endomembranous systems. It's going to help process products for delivery throughout the body. So once you've created a protein or you've created a lipid that you're going to use inside or outside of that cell, it's then going to go to the Golgi apparatus that is then going to process and package those proteins and lipids and other molecules. Once it processes them, it puts them in this little transport vacuole, and it'll take it around the cell, or it takes it to the cell membrane, where it then merges with the membrane and excretes it outside of the cell, so it can be carried to other parts of the body that may need to use it. And so looking here, you see the transport vesicle is going to butt off from the smooth or rough ER. It then goes and joins with the Golgi apparatus, dumping its contents inside. Here, the Golgi apparatus is going to modify these molecules as they move through the chambers. Modified molecules are then going to butt off in the Golgi apparatus to a transport vesicle. Sometimes it's called a Golgi body. Then that vesicle may fuse with the plasma membrane, dumping the contents outside of the cell and delivering them elsewhere in organisms. So in summary, the Golgi apparatus is another organelle within the endomembrane system. It processes molecules synthesized within the cell and packages them to be used outside of the cell. The cell wall is going to provide additional protection and support for plant cells. So the cell wall is going to be found outside of the cell membrane. So if you look at the picture, the cell membrane will be a lighter color, and then the cell wall is going to be outside of it or surrounding it. And so it's going to give strength and structural support. It increases water resistance and provides some protection from insects and other animals that might try to eat that plant. You can see these different connections that will help the plant communicate with other cells around it, and that's the plasmodesmata that we talked about. So all of the cells are interlinked or connected. So in summary, the cell wall is going to be an organelle found in the plant and some other non-animal organisms. This will be things such as prokaryotes, bacteria and archaea. It's going to be made primarily from a carbohydrate in the cellulose, and it surrounds the plasma membrane of the plant cell. The cell wall confers tremendous structural strength on plant cells, gives plants increased water resistance, provides some protection from insects and other animals that might try to eat them. And in plants, the plasmodesmata are going to connect the cells and enable them to communicate each other. Vacuoles are going to be multi-purpose storage sacs within the cell. We talked about in plants, you have a large central vacuole, which contains water, but you're going to have vacuoles that contain other substances as well throughout the cell. For example, the lysosome is going to be a large vacuole that consists of different types of enzymes and acids that'll break things down. So it's going to store nutrients, it can retain and degrade waste products, accumulate poisonous materials, contain pigments enabling plants to attract birds and insects to help them reproduce, and provide physical support, like in the large central vacuole filled with water. The central vacuole can play an important role in 5 different areas of the plant. So like we said, mainly it's going to store water and help with the plant's rigidity and be able to stand, but it also helps with nutrient storage, waste management, predator deterrence, sexual reproduction, and physical support. So in summary, plant vacuoles can occupy most of the interior space of the cell. They appear in some other eukaryotic species, and they function as storage space and play a role in nutrition, waste management, predator deterrence, reproduction, and physical support. Our next is going to be the chloroplasts. These are only going to be found in plant cells. Chloroplasts are going to be the site of photosynthesis within the cell. So this is going to be able to convert sunlight into energy, and it's going to do it into a chemical form of energy, which we refer to as glucose. You can have a stack of these membranous systems. Each one is going to be called a thylakoid, and a stack of them is referred to as a grana. And so these are going to be found inside of each chloroplast. And we'll get more into detail with these when we go into the next unit. So the stroma And interconnected little flattened sacs are going to be called thylakoids. The stroma is going to be like the cytoplasm inside of the chloroplast. This is also thought to be an organelle that was produced through endosymbiosis. So it resembled a photosynthetic bacteria, and they think that it was brought into the plant cell the same way that the mitochondria was. It also contains its own DNA that does not match the cell, and it is in a circular form. And it has a dual outer membrane, which suggests it had a cell membrane. And then it also has the membrane of the cell that it wrapped around as it came in through endosymbiosis. And so here's just a little checklist and a review of the different organelles that we covered, which ones can be found in animals and plants, and their function. So in summary, the chloroplast is the organelle in plants and algae that's going to be the site of photosynthesis. It converts light energy into chemical energy with oxygen as a byproduct. Chloroplast may be originally found as a bacteria that were engulfed by the predatory cell by endosymbiosis. And here's just a breakdown of different eukaryotic organelles. We talked about the nucleus, the nucleolus, chromatin. Nucleoplasm we did not cover, but that's going to be cytoplasm of the nucleus. We talked about the nuclear membrane being a phospholipid double bilayer, so it's technically 4 layers. We talked about nuclear pores. We had cytoplasm, mitochondria, ribosomes, rough and smooth ER. We talked about the Golgi apparatus. It created Golgi bodies, which are the transport vesicles. We talked about the cytoskeleton. Centrioles are only going to be found in animal cells. Remember, they're going to help pull chromosomes to one side or the other during mitosis. We discussed lysosomes. Peroxisomes are going to be enzymes that are found in vesicles or sacs. They're like lysosomes, but they break down different things and help catalyze reactions. These are also going to be very important in breaking down alcohols in the body. We talked about vacuoles, chloroplasts, and cell walls. And our overview again. That's going to end this part 2 of Unit 3 cells.